Today's topic for discussion is topic of plant pathology, and the name of topic is early blight disease of potato. The disease is called as early blight because it occurs early in the season. It occurs very commonly on young plants, and blight, it is a type of symptom in which dead tissue occurs. The affected region shows dead tissue. Such type of symptoms that are called as blight. And potato, it is a very important vegetable crop. Botanical name of potato is Solanum tuberosum. And the early blight disease of potato, it is caused by a fungal pathogen which is known as Alternaria solani. Sometimes it is also called as Alternaria blight disease of potato. Let us see all the details of the disease. Early blight disease is very common in the region where potatoes are grown. Even the pathogen also infects the tomato plant. Especially the leaves of the host plant get affected by the Alternaria solani pathogen. It is of common occurrence both in cold as well as warm regions of India, and throughout the world this disease occurs where the potatoes are grown. The outbreak of early blight of potato during past 5 to 6 years, that is during 1970, 65 to 71, it was a very serious disease in India at Uttar Pradesh. The pathogen causes injury to the leaves. Initially, small spots are developed, and these spots gradually increase in size, and the affected leaves, they fell off. Defoliation occurs. Let us see the symptoms of the disease. Just I told you, the disease occurs especially on the leaves, and when it occurs very early in the season after sowing of the crop. Within 3 to 4 weeks, very small isolated spots are formed on the leaf having pale brown to dark color. The spots are mostly oval or angular in shape, and initially the spots are 3 to 4 mm in diameter. These spots are encircled by chlorotic margin on the margin of the spot that appear yellowish, and that type of margin is called chlorotic margin, and the chlorotic zone gradually increase in size as the spot also increases. That what happens, the infected plant, the lower leaves of the infected plant or the host plant shows infection at the initial stage, because the pathogenic organism which occur in soil, by chance it comes in contact with the lower leaf surface during irrigation or by any other agency, and the leaves get heavily infected. The lower leaves of the host plant get infected at initial stage. The necrotic tissue of the spot, necrotic tissue means dead tissue ka spot. The spot which consists of dead tissue, that is called necrotic spot. Okay, necrotic tissue that shows a specific shape and structure. That spot, the specificity of the spot is concentric rings are formed in the spot. The spot shows concentric rings, and this is the characteristic feature of the spot which is caused by Alternaria solani in the plant. The number of spots on the leaflets or the leaves gradually increase, and the gradual increase of the many spots on the leaf, they occupy most of the area of the leaf, and these spots are generally brownish in color. In severe cases, what happens, the infected leaves dry up and shrivel and droop off, or they fell off from on the ground. When the leaf get infected, that much area there is no photosynthesis occurs where the spot is present. Hence indirectly there is effect of the disease on photosynthesis of the plant. If there is no photosynthesis or decreased photosynthesis, automatically fruit preparation process reduces and very few yield of the crop is observed. Let us see the effect of the disease. How is the effect of disease? The effect of disease on the potato crop may sometimes be considerable. The peak period of attack of foliage blight or the infection on the leaf of potato usually coincide with the time when the plants have begun formation of tubers. The injury to the foliage, the injury to the leaves of the potato, which in extreme cases may lead a premature defoliation of the leaves. Defoliation means the infected leaf fell off on the ground. That is called defoliation. And this defoliation process due to infection of Alternaria solani reduces photosynthetic activity of the plant. Consequently, the tubers formed remain very small or few in number. This reduces yield of the crop. The disease as well integrates rotting of the tubers. Gradually the infection reaches to the tubers through irrigation water or through host tissue, and even the infected tuber shows blight symptoms on which even the rings of the spot are observed. Next, let us see the causal organism. The pathogen which causes blight disease of potato is known as Alternaria solani. It is a fungus. This fungus, Alternaria solani, which causes early blight disease of potato, it is the member of Detromycetes. Detromycetes is the class of fungi in which perfect stage is absent. Hence the group of fungi, this Detromycetes, it is also known as fungi imperfecta. Let us see the structure of the Alternaria solani fungus which causes early blight disease of potato. The fungal body, that is fungal threads, it is known as mycelium. Mycelium, it consists of light brown hyphae, many hyphae together known as mycelium. The hyphae are septate. Septa are present in the hypha, and the hyphae which becomes dark colored at maturity. The dark colored mature hyphae, they enter into the intercellular spaces of the host. What is intercellular space? The space which is present between the cells inside the host tissue. When the pathogen occurs between the cells of the host tissue, that pathogen is described as intercellular pathogen. And later on, this intercellular pathogen, when it occurs between the cells of the host, that produces some small protuberances which absorb food material from the host tissue. And sometimes the fungus penetrate the cells of host tissue and absorb food material. What happens in presence of enough food material to the fungus? Fungus absorbs enough food material and It reproduces by producing conidia. In presence of enough nutrition to the fungus start reproduction inside the host tissue. The fungus produces small fertile hyphae which are erect, which are known as the fertile hyphae known as conidiophores. Conidia producing stalk-like structures means conidiophore. Phore means stalk. Okay. Conidia producing stalk-like structures which are known as conidiophores, which are relative. The conidiophores of Alternaria soliani are relatively short. They are generally 50 to 90 mu in long and 9 mu broad and dark colored, because at maturity the fungus becomes dark colored. Okay, and these Alternaria solani conidiophores, they comes out through the stomata. They comes out through the stomata, and the conidia, which measure 120 to 296 mu in length and 12 to 20 mu in width, and their conidia are dark colored. The conidia which are produced by Alternaria solani, they are very specific. Such type of conidia occurs only in Alternaria. The conidia are beaked. It is having a beak-like structure, pointed structure, and these conidia are muciform. They are broad at the base, pointed at the tip, and these conidia that consist of a transverse septa as well as longitudinal septa. Each conidium is having many cells, multicellular, multiseptate conidium, not cellular, it is a multiseptate conidium is produced, the conidia that which consist of transverse as well as longitudinal septa. Such type of conidium occurs only and only in Alternaria species. The beak of the spore, the beak of the conidium or spore is long, it is septate, and rarely it is branched. Why it is branched? At the time of germination, the elongated beak becomes branched. The conidia are borne singly, but in pure cultures in short chains of 2 or 3. And each conidium develops from a bud formed on the terminal cell of the conidiophore. The mature conidia are detached or released rapidly, readily, and they are carried away by air currents or water or by insect or by any mechanical agency. The conidia are transferred from one place to other. And whenever the conidium gets a suitable host plant that germinate in presence of very small quantity of moisture, and it germinate by producing a small germ tube, and that germ tube infect the host plant and a disease occurs. Let us see how is the disease cycle of the pathogen. Disease cycle of Alternaria solani, which causes early blight disease of potato. This disease is also known as Alternaria blight disease of potato. The mycelium and conidia of the pathogen, they remain on infected plant material, plant residue, or on the ground in the field for long duration. Even after 17 months it can be survive in soil. The source of primary inoculum is the infected plant debris. After harvest of the plant, the infected leaves fell off from the ground, that is infected plant debris, and that is responsible for infection of newly grown crop in the field in the next year. And after germination, this conidia that requires 28 to 30 degrees Celsius temperature for germination, and the germ tube gain entry into the lower leaves of the host plant through stomata. This has been observed by several plant pathologists. The incubation period varies from 48 to 72 hours. And The primary infection usually appears on the leaves of the potato plant within 2 to 3 days under favorable environmental conditions and moisture. Let us see how is the secondary spread of the pathogen. Secondary spread immediately follows through conidia produced on primary spot. What happens when the leaf get infected and the spots are formed? When the spots increase in size and matures, the spores from that spot they are released during due to rainwater or due to irrigation water, and those spores in the same season when they infect to the another healthy leaf, that is the secondary infection. And the mature conidia are readily detached and dispersed through the agency of air currents, water, insects, etc. and a new infection occurs to the adjacent leaf in the same season, that is the secondary inoculum. Let us see the epidemiology of the disease. Epidemiology of the disease: climatic or soil conditions which are unfavorable to the host and thus reduce its physiological vigor that tend to promote the disease. Let us see. The disease spreads and becomes serious when the season becomes with abundant moisture. If there is abundant moisture in the environment that is favorable for the growth and development of the pathogen. And on the other hand, high soil fertility tend to reduce the severity of the disease. If the soil is more fertile, the field is more fertile, field is having more fertile soil in which the crop is grown. So in such soils, in such a healthy soil, the disease never becomes very serious. Okay, and next. Control of the disease. How to manage the disease which is caused by Alternaria solani. How to manage the Alternaria blight of potato. Let us see. What happens? The disease eats soil borne. The infected plant debris that remains in the soil, and that acts as a inoculum for the next crop. Hence, it is very important if you follow the crop rotation, that will reduce the disease intensity. Rotation of the crop. This year you have sown potato, next year you have to alter the crop with other crop. You have to sow other jawar, other crops you have to grow. In the same field, this is the crop rotation. And field sanitation. What is field sanitation? Before sowing of the crop, you have to collect all the plant debris of initial crop, and that you have to burn away from the field. This method is field sanitation, and this also reduces disease intensity in the field. And infected dry leaves and dead stalks of the plant also immediately removed after harvest. That also helps to reduce the disease. And there are several fungicidal chemicals. Fungicides are available in market for the management of the disease. And Bodo mixture is still a effective fungicide, but there are several new fungicides are available in the market today, and those are very useful for management of the disease. And in the earlier books it has given Gnab, Dathan M45 and Bordeaux mixture. There are several fungicides are given, but now these fungicides are not available in the market. Now recently the new fungicides are available in the market that we have to use for the management of the disease. And whenever you use the fungicide, you have to follow all the instructions given on the fungicidal box, because otherwise if you spray high concentration of the fungicide, that is very harmful to the crop. Therefore, the recommended doses of the fungicide is useful for the management of the disease.